This is Rachel Silvio, and this presentation is Glycolysis, Gluconeogenesis, and the role of Aldolase gene expression for Chem 530. Here are the objectives for this presentation. By the end of this presentation, the students will be able to describe the steps of the glycolysis pathway, describe the steps of gluconeogenesis pathway, and compare it to glycolysis. Compare and contrast the different isoforms of aldolase, A, B, and C, and their different expression patterns, and examine how aldolase B expression can be regulated by different transcription factors. Cellular respiration is a three-part process: glycolysis, Krebs cycle, and the electron transport chain. It uses glucose and oxygen to produce energy in the form of ATP for the cells in the body, as shown on the picture in the bottom left of the slide. Cellular respiration also produces water and carbon dioxide. Glycolysis is the 10-step metabolic pathway in the cytoplasm where glucose is broken down into two molecules of pyruvate, a 3-carbon molecule, in a series of steps. Glycolysis consists of the preparatory phase, producing glyceraldehyde 3-phosphate, which is G3P, and using 2 ATP, and a payoff phase, producing pyruvate and 4 ATP. Glycolysis yields a net of 2 ATP and 2 NADH molecules. The reactants of glycolysis are glucose, a 6-carbon carbohydrate, 2 ATP, which is used up in the preparatory phase, converting glucose to 2 molecules of G3P, and 2 NAD+. The final products are 2 pyruvate molecules, which are 3-carbon compounds, 4 ATP, yielding 2 ATP total, and 2 NADHs. These NADH molecules are then used as electron carriers in the electron transport chain in the mitochondria, ultimately producing more ATP using proton gradients across the mitochondrial membrane and the protein ATP synthase. So if we break the word glycolysis up, we have glyco and lysis. Glyco is sugar and lysis is splitting. So glycolysis or glycolysis is the splitting of sugar or glucose in this case. So let's analyze this picture and walk through the preparatory phase of glycolysis. The preparatory phase is the phosphorylation, the use of 2 ATP molecules, of glucose to convert it to glyceraldehyde 3-phosphate, or G3P. So the first reaction here, from here to here, uses an ATP molecule to phosphorylate the glucose and convert it into glucose 6-phosphate, using the help of this enzyme here, hexokinase. The phosphate is attached to the 6th carbon right here. This reaction is not reversible, meaning there is a different enzyme that functions in converting the glucose 6-phosphate back to glucose in gluconeogenesis. Now, the second reaction uses the phosphohexose isomerase to convert this glucose 6-phosphate to fructose 6-phosphate, a conformational shape change. The third step in glycolysis, still in the preparatory phase, also uses a phosphate from another ATP molecule. The only difference between the fructose 6-phosphate in step 2 and the 1,6-biphosphate in step 3 is the additional phosphate group here attached to the first carbon. So now we have 2 phosphate groups. Step 3 is not a reversible reaction, so there is a different protein to dephosphorylate. Now, step 4 uses the enzyme aldolase to split the 6-carbon fructose 1,6 biphosphate into two 3-carbon molecules, which are glyceraldehyde 3-phosphate, G3P, and dihydroxyacetone phosphate. I will discuss aldolase in further detail later in this presentation. So step 5 here uses the enzyme triose phosphate isomerase to convert the dihydroxyacetone phosphate, one of the 3-carbon molecules, into a second glyceraldehyde 3-phosphate, the G3P, so that at the end of step 5, there are two G3P molecules. Steps 6 through 10 are the payoff phase of glycolysis, where there is a positive net ATP produced. Now in step 6, here, two molecules of glyceraldehyde 3-phosphate, G3P, are oxidized into 1,3-biphosphoglycerate by using the two ATP molecules being assisted by glyceraldehyde 3-phosphate dehydrogenase and reducing two NAD plus here into two NADH molecules and hydrogen ions. So step 7 here is the first step in glycolysis that produces ATP. Two molecules of 1,3-biphosphoglycerate is converted into two molecules of 3-phosphoglycerate. Using the phosphoglycerate kinase, this step produces two ATP molecules by taking off the phosphate from the first carbon of each 1,3-biphosphoglycerate and phosphorylating the ADP. So it's producing here the two ATP. Now the next step consists of two molecules of 3-phosphoglycerate being rearranged into two molecules of 2-phosphoglycerate using the phosphoglycerate mutase enzyme. So now it's just rearranged here. Now in step 9, enolase catalyzes the reaction of the two molecules of the 2-phosphoglycerate into two molecules of phosphoenol pyruvate and two molecules of water. Now the last step of the payoff phase in glycolysis, which is irreversible, uses pyruvate kinase to produce two molecules of pyruvate and two ATP from the two molecules of the phosphoenol pyruvate. Overall, the payoff phase produces two NADH, 4 ATP, 2 water, and 2 pyruvate molecules for every glucose that entered the glycolytic pathway. Now let's take a look at gluconeogenesis. If we split this word apart, we see that gluco refers to glucose or sugar, neo means new, and genesis means origin or formation. So gluconeogenesis is the formation of new glucose from pyruvate. Gluconeogenesis is essentially glycolysis in reverse, with a couple of exceptions I will be discussing in more detail later. Now, this process allows the body to maintain blood glucose if glycogen storage is empty. The brain requires at least 50% of its calories from glucose, and red blood cells use solely glucose for energy. But glucose is not always available at the sufficient levels. So our bodies have a process that makes glucose from other compounds, such as lactate, pyruvate, and certain amino acids. This process occurs in the kidneys, hepatocytes, which are in the liver, and enterocytes, which are in the intestines, and maintains the correct glucose levels when fasting or perhaps lacking carbohydrates in the diet. There are 3 steps in glycolysis that are not reversible in gluconeogenesis. So there are some bypass steps and special enzymes, including pyruvate carboxylase, which is the PEP carboxykinase, fructose 1,6 biphosphatate, glucose 6 phosphatase, which is G6P, and lactate dehydrogenase. We'll talk about these irreversible steps and special enzymes in the next slide. Now let's take a look at gluconeogenesis in a little more detail. So the first step of gluconeogenesis is not the reverse of the last step of glycolysis, because that step is irreversible. It actually takes 2 steps here and 2 separate enzymes to convert the 2 molecules of pyruvate to phosphoenolpyruvate, which is PEP. The first part uses the enzyme pyruvate carboxylase to convert pyruvate to oxaloacetate, OAA, which can be seen as an intermediate compound in the Krebs cycle. Now, 1 molecule of ATP per pyruvate is required for the conversion to pyruvate to OAA. The enzyme PEP carboxykinase then catalyzes the reaction from OAA to PEP. This step also requires energy, 1 molecule of GTP for every OAA converted to PEP. Now the next 6 steps here, this pathway of gluconeogenesis, are the reverse of glycolysis. Notice the double arrows. It uses the same enzyme. So we have enolase, phosphoglycerate mutase, phosphoglycerate kinase, glyceraldehyde phosphate dehydrogenase, triose phosphate isomerase, and aldolase. One of those 6 steps, which is the converting of the 3-phosphoglycerate into 1,3-biphosphoglycerate using the enzyme phosphoglycerate kinase, which is this step right here, also requires energy in the form of ATP. And while this is a reversible step to glycolysis using the same enzyme, it produces 2 ATP molecules in glycolysis on the left side, but it needs 2 ATP molecules on the right side in gluconeogenesis. Now, the next step that is irreversible step is when the enzyme fructose 1,6 biphosphatase converts the fructose 1,6 biphosphate to fructose 6-phosphate. Now the next step is the reverse of glycolysis using this enzyme phosphohexate isomerase, just like in glycolysis. And then the last step here is irreversible of the first step of glycolysis. So we're converting the glucose 6-phosphate to glucose using the glucose 6-phosphatase. Overall, the reactants of gluconeogenesis are 2 pyruvate, 4 molecules of ATP, and 2 molecules of GTP for every molecule of glucose. Now, converting the pyruvate into glucose is an exergonic reaction because it uses a lot of energy. Aldolase is an enzyme that catalyzes the reversible split of the fructose 1,6 biphosphate into 2 3-carbon molecules, the dihydroxyacetone phosphate, DHAP, and the glyceraldehyde 3-phosphate, G3P, in the step of glycolysis. So notice these double arrows. Aldolase also catalyzes the reverse step in gluconeogenesis from the dihydroxyacetone phosphate and the G3P to the fructose 1,6 biphosphate, so in this direction. Now, aldolase also functions in other tissue-specific roles, as different isoforms of aldolases have been known to bind to macromolecules such as F-actin, which is important for cellular function and motility, surface adhesins, which assist cells binding to other cells, and vacuolar H plus ATPase, which is important in pumping protons across membranes, altering the pH. Class 1 aldolases are in animals and plants, and they use ketose sugar substrates. Class 2 aldolases are in bacteria and fungi and use metal cations as cofactors. Within the class 1 aldolases, found in mammals, there are 3 different isoforms. Aldolase A expressed in muscle and red blood cells, Aldolase B expressed in liver, kidney, and small intestines, and Aldolase C expressed in brain, smooth muscle, and nervous tissue. When comparing aldolase DNA sequences, human aldolase A and B had a 66% similarity, B and C had a 68% similarity, and A and C had a 78% similarity. All isoforms had similar residues in the active sites consisting of asparagine 33, arginine 42, lysine 107, lysine 146, glutamine 187, serine 271, arginine 303, and lysine 229. But all 3 have different catalytic properties. Aldolase A and B both function in glycolysis and gluconeogenesis, but the function of aldolase C needs a little further research, but could possibly be neurodevelopmental role, as it has been found in fetal rodent brain, and its expression is similar to nerve growth factor induced B factor, so NGF1B. Now, when comparing human muscle aldolase A and human liver aldolase B, their structure is similar with an alpha-beta barrel structure as seen in this picture, and an active site at the center of the barrel. So you see the purple is the beta sheets, and the red are the alpha helix on the outside. So it is likely that the functions of the different isoforms stem from the different structural elements of the isozyme-specific residues, or ISRs, based on different DNA sequences, not necessarily from the small changes at the unliganded active sites. So the differences in the residues, these ISRs, exist in lysine 146, arginine 42, and arginine 303. Since these ISRs function in substrate binding, changes in these locations could change the kinetic properties of the isoforms, thus changing their overall functions. It was found that when ISRs were mapped onto the tertiary structures of each isoform, the ISR clustered into 2 distinct groups, the distal surface patch, or DSP, and the terminal surface patch, TSP, mostly as helices. Researchers have identified 11 ISRs in aldolase A, 12 ISRs in aldolase B, and 4 ISRs in aldolase C. Aldolase B is involved in glycolysis, gluconeogenesis, and other cellular functions. It has been found to bind to the cytoskeleton of liver cells more easily to other tissues such as muscle or brain cytoskeleton. Aldolase B is also expressed in the kidney, specifically the proximal tubule and intestines. Aldolase B gene expression is tissue-specific and subject to hormonal and metabolic regulation. For example, during times of carbohydrate-rich food intake with increased glucose, the liver expresses aldolase B genes. But during fasting, aldolase B is temporarily inhibited. Transcription factors HNF1 alpha which are hepatocyte nuclear factor 1 alpha, and DBP, which is the D site binding protein, are strong transactivators for the aldolase B gene promoter. CEBP alpha, which is the CCAAT enhancer binding protein, is a weak activator. HNF3 by itself does not alter the activity, and HNF1 beta in proximal tubule renal cells and HNF3 in the hepatocytes antagonize the aldolase B promoter by competitively binding close or on the HNF1 site. So I want to go through these 4 transcription factors. The HNF1 alpha is a transcription factor called hepatocyte nuclear factor 1 alpha and is expressed in liver, pancreas, kidney, and intestines. HNF3 transcription factors bind to cis regulatory part in hundreds of genes that encode for the glycolytic and gluconeogenesis enzymes and hormones. HNF3 beta is expressed in the foregut gut endoderm, where the liver and pancreas develop from during development. HNF3 alpha activates glucagon in the pancreas, and HNF3 gamma activates enzymes in the process of gluconeogenesis during periods of fasting and stress to prevent hypoglycemia. The CEBP alpha is the CCAAT enhancer binding protein. The CCAAT sequence is often found in gene promoters. CEBPs are found in hepatocytes, spleen, kidney, brain, and other organs. And the DBP is a D site binding protein and is expressed in the adrenal glands, kidneys, liver, and many other organs with low tissue specificity. There are over 40 disease-causing mutations, one being the hereditary fructose intolerance, identified in the protein coding region of the aldolase gene, some of which impact the transcription factor binding site, decreasing transcription. There was a study done in 2002 that analyzed different transcription factors on controlling aldolase B gene expression. When researchers disrupted the 2 HNF1 binding sites in the enhancer, transcription was stopped. But then when there was a disruption, such as a single point mutation to only one HNF1 site, there was no effect in the liver, but significantly decreased activity in the kidney. Researchers also deleted the CEBP site in the promoter, resulting in no enhancer function in HEP-G2 cells from a human liver cancer cell line. This data suggests that both the CEBP and the HNF1 function is necessary for the expression of the aldolase B gene in the liver. In review, during this presentation, I described the overview of glycolysis and the glycolytic pathway in more detail, including the steps and enzymes required. I also described the overview of the gluconeogenesis pathway and compared and contrasted it to glycolysis, detailing the irreversible steps and different enzymes. I compared and contrasted the different isoforms of aldolase, A, B, and C, and their different expression patterns, and examined the effect of different transcription factors for aldolase B expression. Thank you.